The human body is not divided into neat little tidy subjects. The presentation, what I'll be doing at my end of the room, is going to be organized more along the lines of how the human body actually works. And so when I speak to you about how interleukin-1, interleukin-6 can trigger the proliferation of lymphocytes in a lymph node in response to a viral infection, the next sentence may very well include a discussion of how those same molecules are acting in the brain to impact our mood. And then I may travel back down to the body and talk about some other aspect of the inflammatory response before returning to the brain and talking about how they're impacting your sleep, your reaction time, your memory perhaps. Let's move on now to the first phase of inflammation, and that is the attraction phase. You don't want the infection to go to the immune system, it'll disseminate throughout the body. You want the immune system to go to where the infection is, and the part of the immune system that is initially engaged in this part of the process are the so-called innate immune cells. They are referred to as innate because they are basically ready to go. It takes a little bit of priming, but measured in minutes, maybe an hour. Furthermore, these cells, which include neutrophils, monocytes, those are the ones circulating in the blood, as well as macrophages, which you'll find in fixed tissues, they are basically non-discriminating. They are not specific. In other words, they have no standards. Rather like the royal family in London. Unlike the T cells and B cells, which comprise the adaptive immune system, the neutrophils and macrophages are capable of going after any number of pathogens. So the macrophage that is likely to reach out and latch on to influenza A is likely to reach out and grab streptococcus or influenza B. That's what I mean by they are non-specific. Oh, they do have some minimal standard. Whatever they encounter has to be recognized as not belonging to you. And that happens because it's missing your molecular fingerprint, the one which identifies all of your cells as belonging to you. It's called the major histocompatibility complex, or MHC antigen. So if something arrives in the body and it doesn't have your signature on it, it's basically fair game. Well, not quite yet. After all, the food we eat is not automatically rejected, and that doesn't bear our MHC type. The pathogen has to see no MHC type and be exposed to what is referred to as a danger signal. This would be a molecule that's produced during the inflammatory process that indicates that some damage is associated with the presence of that bacteria or virus. And that will arouse the cells to now go to work. But first they have to get there. The neutrophils, certainly the most prominent cell type in this category, in the bloodstream, has to make its way to where the injury is. Now that's no trivial task, because you have beneath your skin an estimated 60,000 miles of blood vessel, most of it in the form of microcapillary beds. They're so small, so tiny, that only one cell can get through at a time, and even then it's a very tight squeeze. 60,000 miles. I have no idea if that's true. How on earth would you do the experiment? Can you imagine the size of a lab you would need to measure 60,000 miles of something? Enough to wrap around the planet at the equator several times? But you know, I've seen that stated so many times in the chapter on the cardiovascular system in physiology textbooks, that it's rather like the stories you see on the internet. After a while, you think, well, it must be true. Nobody would repeat anything that outrageous. So whether it's really 60,000 miles, as a lot of people estimate, or not, doesn't really matter. We know it's a lot. So how does that cell, that neutrophil, that monocyte, which will later be transformed into a macrophage, that's circulating up in your shoulder, know that it has to get to the injury in your big toe? Well, one of the first things that happens when a tissue is exposed to a microbe and or is physically injured, chemicals are produced, complement proteins, prostaglandins, leukotrienes, C-reactive protein, and other acute phase proteins, which collectively contribute to the containment process. Some of those chemicals are called chemokines. That means they are basically chemical magnets. They will attract the cells that are needed. They get into the bloodstream, and when a neutrophil up in the shoulder catches a whiff of one of those molecules, it follows it down the concentration gradient. So when that neutrophil comes to a fork in the arterial wall, it chooses the path that has the largest amount of that chemokine. And of course, the greatest amount is going to be where it's being produced in the site of injury. But of course, the cell has to get into the injured site. How does it get through the endothelial cells lining the blood vessel wall? Well, there are other chemicals being produced, mostly the prostaglandins, leukotrienes, which are separating the endothelial cells, allowing a passageway, rather like the aisle between these tables, so that the cells can pass into the area of injury. Obviously, an opening that allows a cell to get through is going to permit fluid to leak in as well. And that's exactly what happens. That, by the way, is the hallmark of inflammation.